This session is entitled The Biology of Aging, Advances and Implications. This session will be moderated by Dr. Richard Hodes, the Director of the National Institute on Aging. By way of the format of this session, Dr. Hodes will begin and introduce the members of the panel. He will then give a brief overview of the Institute's research on aging and longevity. Each of the session panelists will examine aging from a number of perspectives. The genomic perspective, for example, from studies that uncover what the gene sequences of centenarians can tell us about longevity, to the role of telomeres in aging and age-related diseases, to the challenge of cellular senescence. Dr. Hodes will then lead the panelists in a brief discussion, and then we'll open it up to the audience. Dr. Hodes, please proceed.
Oh, thank you. Thank you very much for the chance to be here and be moderating what I think should be a very exciting and stimulating session. Laura Carstensen did a fine job, I think, of introducing the topics here. She spoke of issues of average lifespan and of longevity and made some important distinctions. And in talking about the human history, where the focus has been is on the increasing average lifespan of humans over time. As she pointed out quite dramatically, and in fact, James Vopel, whom she alluded to, has shown that over the period of the last 160 years, if you plot average lifespan versus time, there's been an increase of 40 years from age 40 to 80 in average life expectancy over 160 years. That's a slope of 1 to 4, and it's actually, for those of you who may have seen the plot, a remarkably linear curve, although it reflects different things happening over different epochs. Just as an aside, that means for those of you who will endure 8 hours of this session, if I understand correctly, your life expectancy will be increased by 2 hours. So we know a great deal about what has contributed to increases in average life expectancy over time. As far as maximum life expectancy for humans, far less understood. You heard introduced the wonderful character Jean Calment, who died in 1997 at the age of 122. And it's not clear over the time when average life expectancy has been increasing whether maximum life expectancy, longevity, has changed very much at all. We work in humans, we work in animal models. You'll be hearing of the kind of research that approaches this. We certainly have many instances in animal models in which it does appear to be quite feasible by genetic and other manipulations to extend lifespan. We don't know yet whether this will be translatable in humans. Some will say we know, we just don't know when it will occur. It's certainly a provocative question that we'll be approaching here. The National Institute on Aging and the National Institute of Health have been approaching this issue with interest as one very relevant to the health of our national and global population. There are a number of examples of the kinds of initiatives that have been provocative of the advances we've seen over the past years. Perhaps 20 years ago, there was a time, if one looks back that far, when there was far less organized discipline of the basic biology of aging. And one of the exemplary initiatives begun back then was what was called the Longevity Associated Gene Initiative, in which for the first time people who worked in yeast and flies and worms, mice, and in humans were brought together to begin to identify some common pathways, and it's been a remarkably effective initiative since that time. More recently, an intervention testing program has been developed, reflecting the fact that we now are at a point where we can begin to look at interventions or treatments that can extend lifespan in animal model systems. And perhaps the most recent sea change has been the introduction of a term called geroscience, a geroscience initiative at NIH, which recognizes the fact that aging is not only the province of the National Institute on Aging, but is relevant to the missions of nearly all of the institutes across NIH. And so an initiative in which there's been very intensive and committed participation by many of the institutes and institute directors has built upon the notion that there are common events that occur during aging which are relevant to the aging process, but therefore in a derivative way to any and all of the interests and issues of the National Institutes of Health as they relate to health and the quality of life. Today what you'll hear is first some presentations which Al Reese introduced to you already. We'll follow that with a discussion among the group here, and then perhaps most important and provocative of all, we'll open that up to discussion among all of you. So let me now go ahead to introduce once again the speakers who will be up here on the stage and discussants. Scott Devine from the University of Maryland has worked actively in human genomics, trying to understand the ways in which variants affect health outcomes, but now also the parameter of longevity and life expectancy. Carol Greider, who has worked in the area of telomere and telomere biology, recognized, among other things, by her sharing the Nobel Prize to that end, has understood and helped us to understand the way in which telomerase and regulation of telomeres play a role in cell biology and in whole organisms. Interesting, including the interplay and sometimes counterpurpose between the way in which these mechanisms affect cancer, where our goal, after all, is to constrain cell growth, and aging, where at times we want to promote the survival, health, and expansion of healthy cells. And Norm Sharpless from the University of North Carolina, who is trained as an oncologist and has been studying both in cancer and again in aging, the ways in which similar processes, patterns, reflect the response of cells and ultimately organisms to insults which affect both the dysregulated growth in cancer and the dilemma of trying to understand how to best regulate growth to avoid the sorts of senescent phenomena that are counterproductive or dysregulatory, while recognizing that senescence and cell growth themselves must be regulated in the course of normal development and health. With that introduction and taking no more time from what I think will now be an exciting set of initial proposals to you, followed by discussion, I'd like to turn it to Scott for the first of these discussions.
Thank you.